Ladies and gentlemen, thank you for standing by. Our conference will begin shortly. Ladies and gentlemen, thank you for standing by. Our conference will begin shortly. Ladies and gentlemen, thank you for standing by. Our conference will begin shortly.
Good morning and welcome to Oramed Pharmaceuticals Phase 2 NASH Clinical Data and KOL event. Today's webinar may contain forward-looking statements. Various remarks about the company's future expectations and plans constitute forward-looking statements. Actual results may differ from those indicated by these forward-looking statements as a result of various important factors, including those discussed in the company's annual reports and other SEC filings available on the company's website. Please note that the remarks on today's call will be accompanied by a slide deck, which is available in the Events and Presentations section of the company's website. The company will also be hosting a question and answer session at the conclusion of the prepared remarks. Today's event is being recorded. We are all very excited to discuss the newly released NASH data from the Phase 2 study of Oromed's oral insulin candidate. This was a double-blind, randomized, placebo-controlled, multi-center study to assess the safety and efficacy of oral insulin to reduce liver fat in type 2 diabetic patients with NASH. We are fortunate to have with us an outstanding lineup of speakers today, including key opinion leaders joining us to provide commentary on the clinical results in the context of today's NASH treatment landscape. For today's agenda, we will have opening remarks by our CEO, Nadav Kidron, an overview of NASH by Professor Yaron Ilan, Director of the Department of Medicine at the Hebrew University-Hadassah Medical Center. Following this will be a presentation of the Phase 2 data by biostatistician Dr. Oliver Lopez of Integrium. This will be followed by commentary on the study and current NASH treatment landscape by Dr. Ilan and Dr. Joel Neutel, Director of Research at the Orange County Research Center. The formal remarks will be followed by a live question and answer session with Drs. Ilan and Neutel, who served as primary investigators of this study. Both physicians have extensive experience treating patients and conducting clinical research related to the development of drugs for multiple diseases, including liver disease, diabetes, inflammatory bowel disease, and liver cancer. Their bios are included in the slide deck that is available on our website. It is now my real pleasure to introduce our CEO, Mr. Nadav Kidron.
Thank you, Josh. Thank you, everybody who's joining us today. Thank you, the panelists, for joining us today. We're very excited to talk to you today about Oramed and our positive NASH trial results. So just a little bit of as a reminder, Oramed has an innovative technology platform focused on developing oral delivery solution for drugs currently only available via an injection. Oromed's PAD technology enables small proteins, such as insulin, to travel through the gut and into the bloodstream. This method is more natural and should eliminate many of the negative side effects of injection delivery. The oral administration of current injection-only therapies for conditions including diabetes and NASH should offer clear benefits, including increased compliance and safety, fewer side effects, and greater comfort for patients. We have multiple programs in our pipeline with our proprietary lead candidate, ORMD-0801, being evaluated in several clinical trials. We have, as we reported, a strong financial position with approximately $160 million in cash as of the end of Q3. We believe that ORMD-0801 has the potential to be the first commercial oral insulin product for the treatment of patients with diabetes and NASH, serious conditions with a few treatment options. We're very encouraged by the data being presented today and the potential for our therapy to be used in multiple ways in large patients' population. Today, there are roughly $483 billion worth of drugs and vaccine currently delivered via injection, and the global market for drugs to treat NASH is expected to reach $84 billion by 2029, according to industry reports. While we continue to focus on our oral insulin candidate to treat patients with type 2 diabetes, with phase 3 top-line data expected this coming January, about 2 months from today, we are going to have the phase 3 top-line data readout. But the utility in using oral insulin across other diabetes-related diseases, such as NASH, is very exciting. Importantly, we saw no safety issues in this trial as our oral insulin candidate continues to demonstrate an incredibly safe and tolerable treatment profile. We're currently evaluating next steps in this program, and we're looking forward to sharing those plans in the coming months as they mature. At this point, I would like to turn the program over to Professor Yaron Ilan to provide a brief overview of NASH disease. Thank you, Professor Ilan.
Thank you very much, Nadav, for the kind introduction. So in the next few minutes, I want to share with you an overview about fatty liver disease. So as you can see in this slide, fatty liver disease, or the accumulation of fat in the liver, is a global epidemic. It practically affects all countries around the world. And if you look at the United States, there are over 35% of the population in the United States that have fatty liver, accumulation of fat inside the liver, and these numbers are expected to even grow further with the obesity epidemic. In the next slide, you can see the progress from a normal liver to cirrhosis of the liver. So we have a normal liver, and then due to insulin resistance, the metabolic syndrome, type 2 diabetes, and other factors, fat is being accumulated in the liver. And this is termed NAFLD, or non-alcoholic fatty liver disease, standing for the accumulation of fat in the liver in in patients that do not drink too much alcohol. Then in the next step, the fatty liver can move on to NASH, which stands for non-alcoholic steatohepatitis. There are multiple mechanisms, and I will get to this important point in a minute, that lead or contribute to the movement from fatty liver disease to NASH, and mainly inflammatory reactions inside the liver. And then at the last step, fibrosis or scarring inside the liver is developing, and this leads to advanced cell damage inside the liver, along with full-blown scarring of the whole liver and cirrhosis of the liver and end-stage liver disease. In the next slide, we can see the consequences of this process. So again, we start with a normal liver. 30% of the population will move to fatty liver disease. And as you can see, a large proportion of them will move to NASH, cirrhosis, and unfortunately to HCC, or hepatocellular carcinoma, or primary liver cancer. Primary liver cancer is one of the only cancer where we have very limited therapy for. And unfortunately, because fatty liver disease is an epidemic, liver cancer is now the 4th leading cause of death from cancer, because this is a very common disease that affects a large proportion of the population. They just progress from normal liver to fatty liver to NASH, and then cirrhosis, and then to liver cancer. But as you can see from this slide, you don't need to have cirrhosis of the liver to develop cancer. If you develop fatty liver or NASH, you can move directly to developing, unfortunately, this liver cancer. And this is a major concern of health authorities over the last few decades, because when you talk about epidemic that leads to cancer and leads to end-stage liver disease, this is a major burden on health systems all over the world. In the next slide, you can see the fatty liver disease market. So it is expected that the fatty liver disease market will reach around $30 billion within 10 years. And some people even use the number $40 billion within 10 years. And this is because the epidemic is practically spreading all over the world, and there is no treatment. In the next slide, you can see the treatment landscape for fatty liver disease. Unfortunately, despite a lot of effort over the last decade, there are no FDA or EMA approved medication to treat NASH. This disease has multiple mechanisms, which requires multiple approaches. This is just an example of multiple, one out of hundreds of compounds being tested for NASH. And as I mentioned, even those drugs that already finished phase 3 did not reach an approval by the FDA because they were not able to show significant efficacy and relative high safety profile. So what is the unmet need? If we go to the next slide, the unmet need for NASH is first of all for the drug to be safe. NASH is a slow growing process. It develops over decades. A patient who starts a therapy when he's 30 years old will require to take the drug for 40 or 50 years. This drug must be safe. Unfortunately, many of the compounds currently being tested have a lot of question marks with regard to their long-term safety. So you cannot approve a drug that has a safety concern for patients to take it over 30 or 40 years. The next major issue is the fact that, as I showed you, there are multiple mechanisms that affect NASH, which implies the need for combination therapy. If there are so many mechanisms involved, why aren't we reaching efficacy with the current compound being tested? Because each of these compounds target one mechanism. Maybe we can combine some of these drugs together. But then the issue of safety come again. If each of these compounds has a safety concern and you combine them together, the safety is a major challenge for the drug companies. So I think, as I showed you in this brief introduction, NASH is really a major problem and a major unmet need practically all over the world. So I will now turn the program over to Dr. Lopez to present the Oramed phase 2 NASH clinical trial data.
Thank you, Professor Yaron. The title of the study was presented as a double-blind, randomized, placebo-controlled, multi-center study to assess the safety and efficacy of the oral insulin to reduce liver fat content in type 2 diabetes patients with non-alcoholic steatohepatitis. In the next slide, you'll see the introduction and the information about this study. The primary objective is to evaluate the safety of oral insulin in patients with NASH and type 2 diabetes. The secondary objective is to assess whether oral insulin may be effective in reducing liver fat content in patients with NASH and type 2 diabetes. The exploratory outcomes in this study was change in baseline at week 12 in FibroScan median fibrosis, change in baseline at week 12 in FibroScan median steatosis, and change from baseline at week 12 and week 16. Week 16 was post-treatment in HbA1c. Overall, there were 32 subjects randomized across 5 study sites. 21 subjects were randomized to receive ORMD 801 8 milligram BID, and 11 subjects were randomized to receive placebo. Something important to note is that the study was not powered to detect statistically significant differences, and therefore, we're really interested in looking at the patterns in the data. In the next slide, we'll talk about the secondary objective to assess whether oral insulin may be effective in reducing liver fat content in patients with NASH and type 2 diabetes. In the next slide, we see the individual percent change in MRI PDFF levels at week 12. In the table to the left, you'll see the baseline levels for each segment of the liver. In the figure, if I can call your attention to the blue being placebo, the red being those patients on active treatment. This figure shows the MRI percent change for the whole liver, for segment 3 on the left lobe, for segment 6 on the right lobe, and segment 8 on the right lobe. The percentage change you'll see among subjects that took active drug show a tremendous increase, and more subjects on active experienced a decrease in MRI PDFF, and not as many subjects on placebo. In the next slide, we'll see that whole liver subjects on active experience a whole liver decrease in MRI PDFF over 12 weeks. You'll see that difference on segment 3 was quite a significant decrease. In the next slide, we'll talk about the exploratory outcomes, the change from baseline at week 12 in FibroScan median steatosis levels, the FibroScan median fibrosis levels, and the change from baseline at week 12 and week 16 in HbA1c. Here we show the individual change in FibroScan steatosis median levels over time. Presented to you on the left are the baseline levels. These individual changes over time at week 12, we see that predominantly for those subjects on active treatment experienced a decrease over 12 weeks. Very few subjects experienced a decrease over 12 weeks that were on placebo. Again, calling your attention to red being those on active, blue being those on placebo. In the next slide, we see the median change in FibroScan steatosis median levels over time. There was a 29-unit difference between the median level at week 12 for those on active compared to those on placebo. In the next slide, we see the individual fibrosis median levels over time. Among those on active treatment, over half of the subjects experienced a decrease at week 12. Among those on placebo, over half of the subjects experienced an increase in fibrosis median levels at week 12. In the next slide, we see the fibrosis median within each treatment group. Among those on active, though they experienced a median decrease over time compared to those on placebo. In the next slide, we see the mean change in HbA1c levels over time. Calling your attention again to blue being those on placebo, red being those on active. At week 12, there was a mean decrease in HbA1c among those on active and a mean increase among those on placebo, for a placebo-adjusted difference of 0.5 at week 12 at the end of the treatment period. It is interesting to note here that 4 weeks after the treatment period ended, there was still an effect of the active treatment on HbA1c levels at week 16. In the next slide, we see the summary of results. The primary objective of safety was met with no serious adverse events and no difference in incidence rates of adverse events between active and placebo. The secondary objective of reducing liver fat content in patients with NASH and type 2 diabetes, specifically the percent change from baseline to week 12 and MR-PDFF percentage, we saw a placebo-adjusted mean decrease in the whole liver and a placebo-adjusted mean decrease of 10.87 for segment 3 in the left lobe. In the next slide, I summarize the exploratory and other highlights of this trial. The exploratory objective of median change from baseline in FibroScan at week 12, we saw a placebo-adjusted median decrease of 29 for Oromed's product, and a placebo-adjusted mean decrease of 1.1 for Oromed's product. Other highlights consistent with these results: the mean change from baseline to week 12 and week 16 showed a placebo-adjusted mean decrease of 0.5 and 0.58 for ORMD 0801. Lipids showed a placebo-adjusted mean decrease at week 12 also from baseline, consistently across all lipids. Now, I will now turn the program over to Professor Ilan and Dr. Neutel to provide commentary on this Oramed data in the context of the treatment landscape for NASH disease.
Thank you very much, Oliver. So I would like to make a few points about this data. With all the reservations of being a preliminary study, it was not designed to have a statistical significance. There are several up-and-coming trends which are very clear from this data. And I would like to highlight several of these significant trends that come out of the data. The first is the high safety profile, which was already shown in large trial using this compound and was also documented in patients with chronic liver disease in this trial. And as I mentioned before, this is an important factor. While there are drugs to reverse or being tested to reverse fibrosis, you need to remember that the mechanism of action that these patients have, like insulin resistance or inflammatory reactions due to many other causes, do not disappear because we give them a drug to reverse the fibrosis. They are there. We do not change the patient if we give him an anti-fibrotic drug. That means that the drug that we administer for NASH will need to be administered for a long period of time. The second important point is that the drug reduces fat in the liver, and it was measured by two measurements. Both the MRI and the FibroScan, both of them showed reduction of the liver fat in these patients compared to placebo. And the third important factor is that based on the FibroScan data, the drug reduces liver fibrosis, which is the primary endpoint for the FDA NASH trial, within a very short period of time. These patients were treated for only 12 weeks, and we already saw signs of reversal of fibrosis, which is quite remarkable, even if we compare it to all other drugs that are being tested for fibrosis. Most drugs being tested for NASH did not exert an anti-fibrotic effect within 12 weeks. Important to note that there was no change in body weight between the two groups, which is crucial as it supports a direct effect of the drug on the disease, irrespective of the body weight. So while drugs that are being developed for weight reduction are important and are going to be used by patients with NASH, hopefully in the near future, if the phase 3 trials will show benefits, this compound that was tested in this trial, irrespective of weight reduction, had a direct effect on the mechanism of the disease, on the insulin sensitivity in the liver, reducing fat and reversing fibrosis. So obviously, if we look at the mechanism of developing fatty liver disease and NASH, insulin resistant is really fundamental to this process, even in patients who are not obese. And it's important to remember that 20% of the people with NASH are not obese. They are not going to be treated by drugs to lose weight because they are not obese. But they all have a certain degree of insulin resistance, which this compound is targeting. And insulin resistance is an essential variable in the mechanism of the disease. And improving insulin sensitivity in the liver is therefore very important. The fact that the improved parameters in the liver continued during the follow-up further support a drug's biological effect. It shows that we are affecting something in the biology of the disease in this patient. It's important to note that this study was really too short to show an effect on the disease itself. NASH studies now require at least 2 years of follow-up and double biopsies. But the effect that we saw within a short period of time on both fat content and reversal of fibrosis are highly promising. And with that, I'm going to move the discussion to Dr. Neutel.
Well, thank you very much, Professor Ilan, and it's a pleasure for me to have a little opportunity to speak with all of you. And I just wanted to add to what the Professor Ilan has said. I was also one of the investigators on the study, and one of the things that impressed me the most about this compound was the fact that it is clearly the most physiologic way of treating fatty liver, NASH, and for that matter, type 2 diabetes. As you all know, you cannot take insulin orally for the reason that insulin is a polypeptide, and if taken orally, would be degraded in the stomach and made ineffective. What the Oramed 0801 compound has done has encapsulated insulin together with a soybean trypsin inhibitor and EDTA. And when taken orally, the soybean trypsin inhibitor protects the insulin through its passage in the stomach. And when it gets to the upper small intestine, which has a very high alkali content, the insulin is liberated, and with the help of the EDTA is actually absorbed into the portal vein and transported to the liver, insulinizing the liver. And this, I say, mimics, or is more physiologic, because it mimics what happens in healthy subjects, in that in these subjects, insulin is made in the pancreas, transported to the liver through the pancreatic duct, and insulinizes the liver through this mechanism. Now, as you've heard from Professor Ilan, the insulinization of the liver is highly important in the pathogenesis of fatty liver and NASH. Insulinization of the liver increases insulin sensitivity in the liver, and there is a very close correlation between liver fat content and the hepatic insulin sensitivity. So you can see that insulinization of the liver and increasing insulin sensitivity in the liver is a very important part of the management of this disease process. And we've struggled to do this using injected insulin because you have to give much higher doses of insulin in order to insulinize the liver when given by injection. And that, of course, is associated with significant side effects and cannot be well tolerated by the subjects. So you can see that this process is physiologic and is very important in the management of this disease. I think what has been mentioned briefly, but is also important, is that the other changes that we saw in the study were very consistent with what we saw in the liver. There was impressive decreases in A1C in these patients. There was decreases in total cholesterol, LDL cholesterol, and triglycerides, all of which are consistent with what we're seeing in the liver. So that's very important. The other thing, like Professor Ilan said, that I think is critical is in all the studies that we have done using this compound, hypoglycemia has not been a problem. We have not seen any increase in the incidence of hypoglycemia, as you might see with injected insulin. And this is related to the fact that its mechanism of action is through liver insulinization. The fact that there wasn't an increase in weight was also very important, because that is another problem that you see in patients with injected insulin. And also, like Professor Ilan said, I think the fact that there's a slow offset of insulinization in the liver is very important. When we look at patients in multiple studies, once the drug has been stopped and we go into the follow-up period, it takes some time for all the parameters to get back to their baseline levels. So you can see the insulinization of the liver occurs and then persists for some time after stopping the drug. So I think this is a very exciting and novel agent for the treatment of these multiple conditions. Certainly NASH and fatty liver, as we've discussed, and of course also going to be useful in the management of type 2 diabetic patients for the various reasons of being able to protect them from hypoglycemia and yet giving them insulin in a very physiologic manner. So we, I think, are all very excited about this agent. The results, I think, are very important, especially in this group of patients. And so with that, I would like to hand over to our operator so that we can start the question and answer session and see if you have any questions that we can perhaps answer for you. Thank you all so much.
Thank you. In order to ask a question, please raise your hand using your mobile or desktop application and wait for your name to be announced. Once again, please raise your hand using your mobile or desktop application and wait for your name to be announced.
Pete?
Our first question today comes from the line of Robert Lieber. Please go ahead.
Good morning and congratulations on these results. Based on what you saw and knowing that this was not powered for statistical significance, in the next steps and the next trials, how many patients do you think you would need to enroll to show statistical significance? And there was also a mention of a 2-year endpoint. Is that what you would expect for the next step? Or would you look at 24 and 52 weeks? Or any thoughts on the design of the next stages?
Professor Ilan, please.
Sure. Thank you very much for this important question. So this was a phase 2A clinical trial, which was, as you said, was not powered statistically. For a phase 2B clinical trial, it usually requires 80 patients, and we can look at 24 weeks and 52 weeks. The 2 years trial, which requires double biopsy, is only for the phase 3, which usually requires a few hundred patients. So I think for the next step, if the company decides to move forward with, then we will need an 80-patient trial and look at 24 and 52 weeks using the same parameters of FibroScan and MRI as endpoints. We can also consider doing a double biopsy within a year if we really believe that the drug is really powerful, as we see in these preliminary data, to reverse fibrosis. And then a liver biopsy within 12 months may give also an answer.
Okay, great. Thank you very much.
Thank you. Our next question today will come from the line of M. Marin. Please go ahead.
Thank you. Can you give us any additional color on what you're seeing in terms of the competitive landscape with other, potential therapies that are currently in development?
Professor Ilan, please.
So as I briefly mentioned, there are currently hundreds of compounds being tested in phase 1 and 2, and there are several tens that are in phase 2B and phase 3. And each of them is targeting a different mechanism out of the multiple mechanisms that are associated with the development of NASH. And one of the reasons that people believe for the low efficacy of these agents, it's not that they are not working at the right mechanism, it is because that this single mechanism is one of 200 mechanisms that we are aware of, and maybe one of 1,000 that we are unaware of. So we see a very mild effect when you look at the large control trial. Insulin resistance, however, is common to all patients with NASH. It's a fundamental mechanism that starts the process. And as Joel described nicely, this compound that was tested in this trial targets the insulin resistance and mainly the insulin sensitivity inside the liver. And this is one of the advantages of giving oral insulin orally and not injecting it. When we administering it orally, it goes from the gut through a portal vein, which takes the blood from the gut directly into the liver. So the first organ to see the drug is the liver. And then the effect on the liver environment and reducing the insulin resistance, improving the insulin sensitivity, is probably what we see. Now, as I also mentioned, many companies are now discussing combination therapy, and there are even ongoing clinical trials, even phase 3 clinical trials with combination therapy. They say, okay, we have a drug that target mechanism A and a drug that target mechanism B. Let's combine them together. The beauty of this compound, if the results are going to be sustainable in a statistically significant power trial, is the fact that this drug can be combined with any of all the other drugs. It is safe, and it is targeting a fundamental mechanisms that all patients have. So it can be combined with other compounds.
Yeah. And just to add to that, I mean, I think, like Professor Ilan has said, we really are targeting the pathophysiology of this disease at its very root. Many of the other mechanisms work through other mechanisms, or other drugs work through other mechanisms, which are going to create other side effects, and that's become a problem, as we've seen with some of these agents. So treating it directly at the source of the problem is always the best way in terms of efficacy and safety, and I think that's exactly what we're doing here.
Thank you.
Thank you. Our next question today comes from the line of James Malloy. Please go ahead.
Hey guys, thanks for taking my question. On the 8 milligram dose, is that likely the go forward dose? Did you talk about timing for potentially starting a phase 3? And looking at the data, obviously it was a smaller trial, not stat sig. Would you anticipate, had there been more people in the trial, that this would have hit stat sig? The data looked that good?
Professor Ilan, you want to start, and then Joel?
Okay.
Yep. I think the answer is yes. I think if we increase the number of patients per group, as I mentioned, in a phase 2b, we can have 30 to 40 patients per group. It should lead to a statistical significant with a power of 0.8 based on our experience with other similar compounds. I mean, with other compounds for NASH. So the effect that we saw here, as you saw, that the majority of the patients that were treated with the drug compared to the placebo showed a positive effect on the two major endpoints of fat reduction and fibrosis reduction. And based on these numbers, we can anticipate, based on what we know, that with a reasonable power, a group of 30 or 40 patients per drug compared to placebo should be sufficient to reach a statistical significance.
Yeah, and with regard to the phase 3 programs, the 8 milligram dose is the dose that has been selected for the ongoing phase 3 programs. And we have looked at higher doses and found that the 8 milligrams is equally effective, and therefore was selected for the phase 3 program. There are currently 2 ongoing phase 3 programs in type 2 diabetic patients, looking at the drug both as monotherapy and then in combination with other diabetic agents. And those are currently ongoing and done very well. As you heard from Nadav, the first top-line data is expected to be available in the first quarter of next year. And so those are things that we'll see in the not too distant future.
And maybe I'll just add, Joel, another 2 important points. In the first slide, as you saw, we saw a very significant effect on segment 3 in the liver, where we saw a major reduction in the amount of fat in that segment. Now, this is just one of the 8 segments of the liver, but it really proved that there is a biological effect here. Because when you talk about fat reduction in the liver, it's not that something is going down in all parts of the liver simultaneously. Things are moving, the fat is moving. We know that the disease itself is not homogeneous. There are areas where you have more fat or more fibrosis, areas with less fat and less fibrosis. But when you look at a specific segment and you see such a major reduction in the amount of fat in a large proportion of the patients on the drug compared to the placebo, because we are looking to see whether there was a real biological effect, it really gives us confidence that we are looking at a real biological effect. And it's also important to note that none of the other drugs that these patients will receive at the background, whatever they receive for the heart disease or whatever other condition that they have, were kept stable throughout the trial. So they were not changed throughout the trial.
And one other thing, Yaron, that brings to mind, and we've had a lot of discussion about this now in recent months, is that shows the value of the MRI in assessing these patients. Because like Yaron said, the distribution of fat is not uniform throughout the liver. It tends to be in areas where there's good blood supply. And those are the areas that you tend to avoid when doing biopsies, because you don't want to hit a major vessel. And so you can see the value of MRI in assessing this. And I think this was a very useful tool in the study, which gives us a lot of good information.
And maybe a quick follow-up on that, if I could, please. The biopsy has been the gold standard for the trial endpoints. Do you think an MRI is something that you guys could be using in a phase 3, or have to go to a biopsy to prove it? Thank you.
Well, currently, as you said, the biopsy is the gold standard for the FDA, but there's a lot of discussion for the reasons that I pointed out, that perhaps sometime in the future that may change. It hasn't yet. Hopefully it will, but if not, that would still be the gold standard, would be biopsy.
So I
Great,
think
thank you for taking questions.
We still have a lot of questions, but I think I see another 17 questions here. I see another 6 questions there. So I just want to summarize something from the company's side, which will answer some of them. And then what I suggest, giving everybody's time limitations, just feel free to email us, and we're going to email it to either Dr. Neutel or Professor Ilan, or if it's something for Oramed, so we will answer it, but this way we can answer the individual questions directly. I just want to say that as far as the path forward, we want to get to a trial where we can show statistical numbers. We're working something with the third party, and as soon as things are maturing and we can go public with things, we obviously will update the shareholders in accordance. So that's why we don't want to talk too much about the exact next steps in this point in time. So I really want to thank everybody for attending this morning, and the panelists as well. We appreciate your continued interest in Oramed. We especially would like to thank Dr. Ilan, Dr. Lopez, and Dr. Neutel for joining us on the call. There is going to be a link to replay the webinar, and the slides will be available on our website, oramed.com. Also, anybody who doesn't get the updates directly from Oramed, I highly encourage you to log into oramed.com, put your email there, and then as soon as we have more things, we'll be happy to send you an email, and you'll be notified of that. We're really looking forward to updating you on our progress, in particular, as I said, our phase 3 trial results in type 2 diabetes coming up in 2 months from today. Have a wonderful day, and operator, you may disconnect.
Thank you.